Today I have two questions related to the positive sense and negative sense RNA. Positive sense and negative sense RNA, you know that related to the viruses. I'm talking about the viral RNA. Uh, as you know that some viruses are DNA containing, some are RNA containing. So I'm talking about the RNA containing viruses. And in case of the RNA containing viruses, there are two possibilities: positive sense RNA and negative sense. Negative sense is also known as antisense RNA. And the second question is, why must negative SS used for single stranded RNA be converted into positive RNA with RNA viruses? So I will explain the first question, then you will get the answer to the next question easily. So first of all, which are the most popular viruses, and you are familiar with those viruses are those like influenza virus, like which is flu virus, and influenza measles virus, rabies virus. All these viruses are those which contain their genome in the form of negative sense RNA. Must keep it in your mind. They are negative sense RNA. Like you can see here, the example of this is the flu virus or influenza virus. And this virus, you can see here, contain its RNA. And this RNA is the negative sense RNA. Is it clear? This RNA is negative sense. And here on the opposite side, you can see the next virus, which is the polio virus. Now, this polio virus RNA is positive sense RNA. And there are other examples of positive sense RNA, like you can say the best example of the positive sense RNA, polio virus, along with the polio virus, coronavirus, and a number of other viruses which are RNA positive. HIV virus is RNA positive virus, and but HIV case is different because HIV contain two strains of the RNA, but polio virus contain the one RNA strain, which is the positive sense. This is positive sense RNA. So poliovirus is also known as enterovirus. Enterovirus, you know that entero is the word used for the intestine. So this virus, poliovirus, actually you know that enters through the gastrointestinal tract, means first infected the intestine. So that's why this is known as enterovirus. Yeah, enterovirus. So this is positive sense, and the flu virus is negative sense. Now the difference between the influenza and the polio virus is clear. Which one is the antisense and which is one is a sense. Now I'm moving towards the question, the difference between the two. In case of the, must keep it in your mind, negative sense RNA, must keep it in your mind, is that type of RNA which is first converted into positive sense RNA when enter into the host cell. You know that viruses can't live without their host. They are obligate parasite. So when they enter into the host cell, okay, in the host cell, this RNA negative means the antisense convert into a sense RNA, means the positive RNA. Because why it is converted, I'm telling you here, explaining this, that negative sense RNA is converted into positive sense RNA because this positive sense RNA act like the messenger RNA. And do you know what is the role of the messenger RNA? Messenger RNA form the proteins. We all know that, undergo the translation mechanism. So this positive, if we talk about the positive, why the negative convert into positive, the reason is that the positive RNA, positive RNA, okay, this positive RNA is just like the, you can say that this is messenger RNA, and this messenger RNA directly form then protein under the process of translation. Okay, so if you say that it directly form the protein, तो just keep it in your mind, ये कैसे है, जैसे कि आप ready to cook है, यानी फ़ौरन से जो मैसेंजर आरएनए है वो प्रोटीन बना लेता है। तो अब जो पॉजिटिव सेंस आरएनए है वो तो मैसेंजर आरएनए की तरह एक्ट करता है एंड फॉर्म द प्रोटीन्स, सो देयर इज़ नो नीड टू कन्वर्ट इनटू अनदर आरएनए। अब रीज़न क्या है? यू नो दैट कि इन द host cell ribosomes are present. So ribosomes actually ribosomes, you know that they form the bond with the messenger RNA. Actually ribosome detect the messenger RNA for protein synthesis ना, ठीक है। तो क्या है कि राइबोसोम डिटेक्ट मैसेंजर आरएनए, सो पॉजिटिव आरएनए इज नो नीड टू कन्वर्ट इनटू अनदर आरएनए। पॉजिटिव आरएनए मस्ट कीप इट इन योर माइंड, क्या है? दिस इज द मैसेंजर आरएनए। बट इन केस ऑफ द नेगेटिव आरएनए or antisense RNA first converted into messenger RNA, and reason is that messenger RNA is actually detected by the ribosome. This negative RNA is not detected by the ribosome for protein synthesis. So the reason is the ssRNA means the negative RNA converted into the positive RNA first and then form the proteins. Is it clear? And how? Now the point is कि कैसे नेगेटिव आरएनए कन्वर्ट होता है पॉजिटिव आरएनए के अंदर, ठीक है। यानी एंटीसेंस सेंस में कैसे कन्वर्ट होता है? इट नीड अ एंजाइम विच इज़ नोन एज आरएनए पोलीमरेज। आरएनए polymerase, which is also known as RNA dependent RNA polymerase, means RNA dependent RNA polymerase, RDRP. This is also known as RDRP. This because this और ये जो RDRP है, this is present in the where in the virus for the conversion of the negative sense, means the antisense, into positive sense, means the messenger RNA. So they have RDRP already for the conversion of negative into positive. Is it clear? So this positive then combined with the ribosomes and form the proteins. Clear concept? Okay. So in case of the positive RNA like poliovirus RNA is the positive sense, so there is no need to convert into another form. So this positive act like the messenger RNA and then it form the proteins when they attach with the ribosome in the cytoplasm of the host cell. So this is the basic concept related to the positive sense and the negative sense, means the antisense and sense. So that's why the negative convert first into positive, because the positive is like the messenger RNA, behave like messenger RNA, and it is detected by the ribosomes of the host cell and form the proteins for the survival, for the reproduction, for the replication of the RNA. You know that RNA consists of the protein coat and the genetic core. So the genome actually replicate, reproduce in the cell and also make its protein as well for a complete RNA particle. So this is the concept. If you need more lectures, you can contact me for the online class. Thank you.